Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Nicole Wakes from Abzena.
Thank you very much for the introduction. So I'm Nicole Wakes. I'm a group leader in the cell line development group in Abzena, and I'm here today to talk to you about how we accelerate timelines by integrating our cell line development and manufacturing programs. So Abzena is a UK and US-based partner research organization for the integrated development and CGMP manufacture. We possess leadership, experience, technical expertise, quality systems, capacity, facilities and equipment necessary to provide the services for cell line development and manufacture. We have over 17 years of experience based in our UK and US sites. In the UK, we're based in Cambridge, where we cover everything from discovery through to design and developability of the molecule, including the cell line development. And in the US, we have presence on the east and west coast in San Diego and in Bristol, Pennsylvania. And both of these sites are involved in the GMP manufacturing of our biologics, as well as the chemistry side and ADC manufacturing in Bristol. So how do we accelerate the timelines within Abzena? So by coming to Abzena, we will develop your cell line through to research cell bank, and following that there will be an expression stability study. We can actually transfer the RCB at the time of generation to our sister site in San Diego, allowing that expression stability study and the GMP upstream and downstream work to start in parallel. But if you are to go to a third party, you would have to wait for the completion of the expression stability study before you can move into the GMP production stage. So by doing things in parallel at Abzena, we are able to save around 4 to 5 months of a standard timeline. So what does the CLD workflow look like? So the variable regions will be codon optimized for your antibody and cloned into an expression vector. This vector will be transfected into our composite CHO cell line, and this cell line is a licensed, royalty, and milestone payment-free cell line. We will generate up to 6 stable pools, and these stable pools can be assessed for productivity in a small-scale batch culture from which material can be purified to support further downstream studies. Selected pools will then be seeded into semi-solid media, and the resulting outgrowth colonies will then be screened and picked on the ClonePix2 system into a liquid medium, which we can take into a shaking environment to assess for titer. At this stage, we have the option of looking at critical attributes for the molecule, be it SDS page for intact molecule, antigen binding, or other analytics as required. Following this, selected clones will be subcloned by limited dilution cloning with visual assurance on the Salentum cell metric to assure monoclonality. The monoclonal wells will then be expanded and screened for titer, again in a shaking environment, before selecting the top 24 clones to assess them for growth and productivity in a bioreactor environment such as the Amber 15. The final clone selection for RCB will be based on the titer as well as growth characteristics, and again we can perform analytics and bioassays as required at this stage to enable that final selection for the research cell bank. We will generate a GMP compliant RCB, which will be tested for mycoplasma and sterility at an external vendor. And at this point, we can then transfer that RCB to our colleagues in San Diego, ready for the next stage. Whilst we are doing the transfer, we will start the expression stability study, generally a 60-generation stability study, and as I said, that will go on in parallel to the initial work startup in San Diego. At any point during this workflow, we can also produce gram quantities of purified protein for any additional proof of concept studies to expedite formulation and analytical method development. So we use the Amber 15 for our clone selection. Excuse me, sorry. This is a widely used model for stir tank reactors, and we have used it for the last couple of years within Abzena, and it mimics the classical bioreactors, allowing us to select the best clones to take forward for research cell banks. The next slide just shows some titer data for the cell lines here at Abzena. On the left-hand side, we have our composite CHO in the blue, so this is just a shaking environment, and we routinely see titers up to about 1 gram per liter for our clones. When we take those into the amber, we then see an increase in titer in the bioreactor environment up to about 3 grams per liter. We've also got a huge amount of experience in other molecules such as fusion proteins, as well as biosimilar and other more novel molecules. Again, alongside our CHO platform, we do offer mouse myeloma platform as well, which we predominantly use for our biosimilars. So upon completion of the initial RCB at Cambridge, we will then initiate the tech transfer to our colleagues in San Diego. Overseeing this whole procedure will be a single project manager. They will be involved in that project right from the start of the cell line development through to the GMP production. Alongside this, we will generate a document transfer checklist to provide to our colleagues in San Diego. This will have all of the necessary information to ensure a seamless transfer of the cell line into their facility. Included in that will be our CLD and stability study reports, as well as the sterility testing certificate. What advantages does this offer to stay with a single provider? So we have familiarity with that cell line. Our platform processes are the same. Our equipment is compatible across both sites. We have the same template. And an additional thing that we will do alongside that is we will provide a CLD scientist to transfer over to San Diego to be on site when the initial work starts at San Diego, again ensuring that seamless integration between Cambridge and San Diego. So once the cell line has been transferred to San Diego, the selected lead clone will be subjected to master cell bank generation. Initially, we will do some process development on a stable pool or a final clone to select that final clone. Then we will generate up to 300 vials of that master cell bank, which will have all the associated testing as per the ICH guidelines, and this is then stored in a secure facility within Abzena. So the processes within Abzena are robust and scalable, and they are suitable for a clinical production system. So initially, the selected clone will be expanded in shake flasks before taking forward to the Amber 250 to do some DOE experiments to optimize and develop the process, and then we will progress from there into 2-liter scale up to 200-liter scale in the stir tank reactor to optimize further and generate our development runs before doing a confirmation run at the larger scale. The downstream process development will be performed from material from these bioreactors by means of an overlapping approach to that upstream and downstream experiment, supporting that expedition of the timeline. So all of this is all done with single-use technology, so this provides a cost-effective production of the molecule. So the 50-liter run is used to seed for the 500-liter run, and the 500-liter run will then provide material for that engineering or tox study. There is also options to include stability studies at this stage as well, and the GMP production then will be initiated around about the time of the final release of the MCB and the completion of the virus clearance studies to provide the safest possible technical solution, and then this will constitute the clinical supply for first in man. On the far right of this slide, we've included a 2,000-liter bioreactor. We are taking delivery of the 2,000-liter in the first quarter of next year and hope to have that up and running by the summer of next year. So again, sort of downstream, the systems we use there again are all disposable flow paths, and again this allows huge amount of flexibility and a very efficient turnaround time. So from the ACTA step through to the viral filtration and formulation step, all the way through to fill finish. Although we ourselves currently do not provide a fill finish service, we have a preferred selection of vendors which are our go-to for this particular service. So this slide provides a very top-level overview of the technical development plan that will be provided to the client for the entire project. We have the critical path shown in red, assuming the tox batch and the GLP tox study are on the critical path. And at the top of the slide, we have the cell line development study shown there as well. So we have milestones in there for the MCB construction and release, banking of the initial reference standard, filling of that toxicology batch round about the month 11, and then the final filling of the drug substance batch round about month 14 and its subsequent release. So what this shows you is that we can achieve a significant time saving of up to about 5 months compared to a multi-vendor approach, and this is also facilitated through the early start of the formulation and analytical method development, which can start before the cell line development process has completed. So by having a combined global analytical workflow, we provide a significant reduction in risk when transferring the cell lines due to the harmonized methods, instrumentation, and documentation that I have talked to you about today. So we have very, very structured communication channels, and this ensures that we have effective information transfer. And while a lot of this information today has been about regular vanilla antibodies, we do have a huge amount of experience as well with multiple classes of biological molecules as well. So in summary, by selecting Abzena as your partner research for your molecule from cell line development through to manufacturing, we will reduce the operational risk, and we can achieve this by having a single project manager and scientific leader to ensure that seamless integration all the way from cell line development through to GMP and keeping things on track. As I've said, the utilization of the latest automated high-throughput technology to rapidly determine the optimal process conditions over a wide range of possible parameters, the use of the Sartorius platform all the way from the early stage within cell line development through to currently 500 litre and early next year 2,000 litre at San Diego. Again, this seamless integration reduces the risk and process variability associated with different equipment from different vendors, but also reduces the number of repeat runs that may be required. Backing all of this up, we have our quality systems, because this is of utmost priority behind all of this, and we have a single QA team who oversee all of the QA aspects of the project, and they have a very strong working relationship with the client team. And then also, again, by opting for a single vendor, we reduce that financial risk. So the integrated solution allows a lot of removal of that white space in the capacity area, leveraging of efficiencies by using a single project team that are very, very familiar with that project and with the client's requirements, with no duplicating of resources. And then this allows a much more seamless and financially efficient transfer of activities to our sister site rather than using multiple CROs. So that concludes the presentation, so I will hand back for questions.
Great, thanks. So the first question is, how does this acceleration differ from accepting the risk of transferring without an expression stability study?
So as I said, we can initiate the expression stability study whilst we are starting the process optimization on either a single clone or a number of clones, so that we are straight into scale-up as soon as we have selected that stable clone. But also early on, we have started analytical method development and formulation studies. So again, we have reduced timelines there.
Okay. And there's a couple of questions. Would you have any experience with having a product quality from the pool that's different from the clones?
So what we will see in the clones, say for example, if we're looking at a glycan profile, we'll always see different between that and the clones is the difference in perhaps amplitude of different glycan profiles. So if you're looking for a particular quality attribute, if it's not present in the starting pool, then it won't be there in the final clone. But generally, there isn't that much difference. It's just the amplitude of various attributes.
And what kind of CHO do you use as host cells?
So we have a CHO DHFR based system.
And what are the criteria for cell line stability?
So we use sort of the sort of industry standard for cell line stability. So this is based on expression. So we'll take a cell line out to generally 60 generations, and the expression will be sort of plus and minus 30% from the original starting titer.
Okay, and I think we have time for just one more question. If we didn't get to your question, those will be passed directly to Nicole, and she can follow up with you individually. So the last question is, do you perform design of experiments?
So we can do that for the AMBER 250 system. So yes, that is an option.
Okay, thank you very much, Nicole.
Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We invite you to fill out the survey at the end, and we look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.